Regenerative strategies, cell-free approaches, cell-based approaches. Cell-free approaches, the currently employed strategies in regenerative endodontics to treat immature teeth with pulp necrosis constitute a good example of cell-free approaches. These procedures depend on stem cell transfer from the apical tissues into a disinfected root canal system. This transfer of cells has been commonly achieved by mechanically releasing cells from the apical tissues along with bleeding. The blood containing the cells also work as a fibrin scaffold, favoring tissue formation. Another clinically used approach is the use of platelet-rich fibra, plasma or platelet-rich fibrin as scaffolds. These two natural scaffolds contain various chemotactic factors capable of recruiting undifferentiated mesenchymal stem cells. Cell-based approaches. Cell-based regenerative strategies will include the transplantation of cells to the site of injury in tissues to promote regeneration. Clearly, such cells should be of the highest good manufacturing practice quality and ideally should be from autologous sources to minimize any risks of immature rejection. These requirements and the necessity for appropriate clinical facilities to handle the cells provide a number of challenges, and in the current context of endodontic practice, cell-based therapies are only likely to be undertaken in a hospital setting. As well as the direct transplantation of the cells, these cell-based approaches may also involve the application or local release of the signaling molecules and include the use of cell matrix or scaffolds delivery system to inject a scaffold with chemotactic factor into the root canals. This approach is called cell homing, as cells are attracted to the scaffold along with supported blood vessels in a progressive manner instead of being abruptly delivered to an avascular space. The cell homing approach can be applied in a cell-free or cell-based approach. Cell homing. In the context of tissue regeneration, cell homing consists of two distinct cellular processes: recruitment and differentiation. Recruitment refers to directional cell migration, including that of stem or progenitor cells to the site of tissue defects. Pulp and dendrin regeneration will not be possible unless mesenchymal stem or progenitor cells with the capacity to differentiate into multiple cell lineages that form pulp and dendrin are recruited into the root canal. So this shows regeneration of pulp tissue after autologous transplantation of mobilized dental pulp stem cells with GSCSF in pulpectomized teeth of young dogs in day 14, that is A and B, and regenerated pulp tissue. B is the odontoblastic cells lining to newly formed or osteotendin or tubular dendrin along with the dendrin. C is the immunohistochemical staining of BS1 lectin. D is the immunohistochemical staining of PGP9.5. Mechanism of revascularization. According to Shan, the possible mechanism by which the process of revascularization takes place are as follows. A few vital pulp cells remaining in the apical end of the root canal might proliferate into a newly formed matrix and differentiate into odontoblastos. This may happen under the influence of the cells of Hardwick's epithelial root sheath, which are quite resistant to destruction even in the presence of inflammation. A tubular dendrin is laid down by the odontoblastis at the apical end of the canal and on lateral aspect of dendrinal walls of the root canal. This leads to apexogenesis and reinforcing the strengthening of the root respectively. Continued root development could be due to the multipotent dental pulse stem cells which are present abundantly in immature permanent teeth. Stem cells in the periodontal ligament can proliferate and grow into the apical end and within the root canals. They may deposit hard tissues both at the apical end and at the lateral root walls. The evidence in support of this hypothesis was presented by documentation of cementum and sharpest fibers in the newly formed tissue. The next possible mechanism of the root element could be attributed to SCAP or to the bone marrow. Instrumentation beyond the confines of the root canal to induce bleeding and also transplant mesenchymal stem cells from the bone into the canal lumen. These cells have extensive proliferating capacity. The blood clot in a rich source of growth factors such as platelet-derived growth factor, vascular endothelial growth factor, platelet-derived epithelial growth factor, tissue growth factor. This could play an important role in regeneration. Clinical procedure.